Welcome everybody to I guess it's the September edition of the IPM Hour. Our guest today is Elizabeth Pringle, an associate professor in the Department of Biology at the University of Nevada, Reno. She got her bachelor's in environmental science from Harvard and her doctorate from Stanford, so didn't overachieve there or anything. And her research interest is the evolutionary ecology of multi-species mutualisms. And I had the good fortune to go out and hang out with her for a while and talk about the project. So, but now she gets to present the scientific side of it. So, Beth, all you.
Great, thank you. I'll just share my screen.
Please.
All right. So this project is looking at whether we can use conservation biological control in arid agroecosystems like we have here in northern Nevada to maximize biocontrol specifically of aphid pests of alfalfa. And the idea here is, I think last IPM hour was focused on biocontrol, right, is to look at the potential predators of alfalfa aphid pests and to see if there are elements of the landscapes that surround alfalfa fields that we can try to maximize in order to maximize the benefits of that suppression of the pests by their predators. I wanted to say just at the beginning here that this is really a collaboration between me and the graduate student that I recruited to work on this project, Anson Call, who couldn't be here today. And in some ways, he would be better at describing some of the things that I'm going to talk about today than I am, but I'll do my best. And Anson really took some of the initial ideas from this project and improve them dramatically in the ways that I'll talk about, in ways that really we learned a lot more from our investigation of the surrounding habitats in these alfalfa fields than I had expected that we would. So we know that semi-natural and natural habitats occupy the space between and around fields in lots of agroecosystems. And in particular in crops like alfalfa, which have alfalfa hay crops, which have these periodic harvests, one potential problem is that as the fields get harvested, any predators or biocontrol agents that were in those crops disperse away during those harvest events. And the potential then to sustain populations of those predators in the landscapes may depend on these surrounding habitats, and that could be other crops or other alfalfa fields, or it could be these semi-natural and natural habitats. And so this kind of thing has been shown in other systems convincingly. For example, in this wheat crop in Europe, where you have these hedgerows of different kinds of flowers, that there can be maintenance of biocontrol agents in these neighboring kinds of habitats. So some of the big questions here that this work addresses is what makes a good predator refuge, and how accessible does that predator refuge have to be to the field? So specifically, what's the spatial scale of relevance for predator dispersal, and how close do those habitats need to be to the field to exert effective biocontrol? Another is whether we can assess and what the best methods are for assessing the quantity and quality of these types of refuge habitats. So here is another meta-analysis that shows that the proportion of cultivated land is negatively associated with the amount of natural or biological pest control of aphids. And this was among all the studies that have looked at that so far, mostly, as I'll talk about, in mesic systems. So we were really interested in how this might apply to more arid systems like those that we have here in Nevada. So the mixed but generally positive evidence so far about the effects of semi-natural habitat on biocontrol is that those habitats have a positive effect on predator populations and therefore on biological control. But a lot of these studies, or the vast majority of these studies that we've reviewed for this work, have occurred in more mesic systems than what we're working in here. A lot of studies coming from Europe, where you can actually see in this picture that incorporates vegetation index, and basically where it's more green here, it means that it's wetter. A lot of the very nice looking verdant habitat is actually outside of the cropped areas in these kinds of systems. In Nevada, by contrast, what we get when we do alfalfa farming are these areas of lots of plant growth, very verdant, and then we have our natural habitat is the sagebrush ecosystems or greasewood ecosystems outside of the fields, and you can really see features of the landscape that have to do with water. When you add water to the system, you get lots more plant growth, and potentially you also form refuges for potential biocontrol agents. So here we're seeing a river that's going by one of our field sites in Fallon, Nevada, and you just see what a difference that makes to the vegetation. I wanted to know that the reason that I got interested in this was kind of by chance. I arrived in Nevada wanting to work on milkweeds and started looking for milkweeds around our university here in Reno, and found that a lot of the milkweeds, we're interested in milkweed monarch interactions and milkweeds as systems for understanding plant chemistry and insect interactions, were found along irrigation ditches in alfalfa growing districts. And that got me thinking about how different these communities are along these canal zones where we basically added water to the landscape, therefore added weeds, some native, some non-native, a variety of different plants that wouldn't otherwise be able to exist, at least at such high densities, in the sagebrush natural ecosystems that are surrounding these fields. So the way that I had originally pitched this work to the Western Integrated Pest Management Center was to think about the effective landscapes at basically a 1 kilometer scale around the fields, and define what kinds of landscape cover there were in that radius manually. So to say, okay, I can digitize this map of the area, and I can say that these are alfalfa fields. I can say that this is a riparian, system following this river, etc. And then try to correlate some of those landscape features with aphid and predator densities, and also with aphid biocontrol. But what Anson really added to this project was his insight that we could use remote sensing, and specifically some of these newer technologies that are just becoming more available and more affordable. What we used was imagery from the Planet Dove satellites that are essentially have all been launched on SpaceX rockets. And we can buy imagery at a 3-meter resolution scale for our field sites here in Nevada for a fraction of the price and easily available than they were a decade ago. So we used our money from the grant to buy about $50 of imagery, and we get this kind of resolution going around each site that we picked to work at. And then we could try to use that imagery to identify features of the landscape more precisely than we would have been able to otherwise. Additionally to that, those images that we got from remote sensing, we also added some detail from other sources, including from the USGS hydrology dataset, in order to try to look specifically at some of our hypotheses. For example, that there would be an effect of these canal systems in flood irrigated alfalfa because of the potential for those canal systems to support these weedy populations that might be of interest as refuges to predators and also potentially to pests. So what Anson did here was to map the stream and canal paths. Again, this is from our site in Fallon, Nevada, which has some 360 miles of additional canals that have been built for flood irrigation of alfalfa, onto our Planet SuperDove imagery, so that we had a good sense of what was going on at a very fine scale in the landscape around the sites that where we were working. All right, so our goal here was to assess biocontrol of aphids. Aphids are a consistent, persistent alfalfa pest, sometimes some years and some fields a worse pest than others. But they've also long been understood that predators could help control these pests potentially better than some of the pesticides that are available, which are not aphid specific, and of course, which aphids can develop resistance to over time, as well as other pests because they're not aphid specific. So our question here is, can we maximize the beneficial effects of the predators that might control aphid pests in alfalfa? The so-called alfalfa aphid complex here in the West comprises 4 species. These 2 species in the genus Azothosiphon, pea aphids and blue alfalfa aphids, which are quite difficult to tell apart actually, but are very closely related to one another. Therioaphis, which is a genus of the spotted alfalfa aphid, and Aphis, which is the genus of the cowpea aphid. There are quite a number of predators suggested in the literature to be important to alfalfa aphid biocontrol. Some of them better tested on their effects than others, so we sought to include all of those that have been suggested in the literature to potentially be important, and that included pirate bugs, spiders, ladybugs, big-eyed bugs, parasitoid wasps, and damsel bugs. And our questions in this study were 4-fold. We first wanted to understand a little bit about aphid and predator density variations seasonally. So again, it's been suggested in the literature that the primary times of year at which aphids might be a problem in our system was in the spring, early spring and fall. And so we wanted to know the extent to which these predators and the pests were varying seasonally, and how that might affect our understanding of biocontrol. We also wanted to know in our system which predators of those 6 categories, taxa that I had mentioned on the previous slide, actually suppress aphids. And then we wanted to get into this question about the semi-natural natural habitat, about what kinds of land cover types are associated with high aphid and or predator abundance and biocontrol, and also what the relevant spatial scale was to consider. Are we talking about something that's happening very close to fields or something that's happening on a broader landscape scale? I believe that this is an AI generated image when you put in ladybug. All right, so our sampling design was to incorporate 4 of the alfalfa growing regions, major alfalfa growing regions in northern Nevada. So we are located here in Reno, and we were working in Minden, Yerington, Fallon, and Lovelock. For 1 farm at each of these sites, and then 3 fields at each farm. And then we chose 3 plots in each field and averaged all of our data within plots. So that's what that looks like at a single site. Minden, we chose 3 fields, each at least 1 kilometer apart, and then we have 3 plots within each field. And within each of our plots, then we had a subplot of 3 treatments that I'll talk about here in just a second. So we had 3 treatments in the subplots within each plot, within each site, within each farm. So the experimental design was to try to tease out the actual effects of biocontrol on these aphids. So although we can do a lot with correlations and regressions, trying to understand sort of which predators are associated with aphids, to actually understand whether predators are suppressing aphids, we have to do some kind of experimental manipulation, because in general, those predators that are going to be best at suppressing aphids are also going to be positively correlated in their densities to aphid densities. So the way that we attempted this was to have 3 kinds of subplots, 3 treatments here. We have a control that's just an open plot. So we just marked the edges of a 1 by 1 meter plot. We have what was supposed to be the full predator exclusion, where we have netting covering this whole 1 by 1 meter plot, and then it's dug into the ground so that predators can't crawl up into the plot. And then we seeded those and the other 2 experimental plots with aphids. So the idea here was that the aphid populations would grow up in the absence of predators, and we would be able to see sort of how exponential aphid population growth looked like in the absence of those predators. And then this was supposed to be a sham treatment, just controlling for the effects of that cover on the predator exclusion. What ended up happening was that this plot was terrible at replicating the population dynamics of aphids in the broader field by simply seeding that plot with just a few aphids, we were not replicating, especially in the spring, what was happening with this sort of rapid aphid population growth in the field. So we ended up excluding those plots entirely, and what we found instead was that this sham control treatment actually ended up acting as a predator attractor. So somehow that white mesh out in the field attracted predators. We were able to look at which predators actually had increased populations in the plot on top of these, and use that as our experimental treatment to compare the controls instead. So to set up the experiment, we initially, using a modified leaf vacuum, sucked all of the arthropods off of the 1 by 1 meter plot, set up our experimental treatments, left for 2 weeks, came back, and then re-vacuumed all of the plots to compare the arthropod densities in all of those plots. There was then a long period of time in which Anson and other folks in the lab were involved in IDing all of these thousands of arthropods that we got out of our plots, and in trying to determine the densities of the 4 different species of aphids, as well as of those 6 different taxa of predators. One of the other features of our design was just to think about a little bit in choosing our fields about the landscape context of each of these fields, because again, the idea was to go on into asking at what spatial scale and what types of land cover were associated with aphid pest suppression. So we chose fields that had slightly different landscape contexts within each site. And so, for example, here you can see the field margin here and here, and then the kinds of sort of bare ground and structures and or sagebrush greasewood type habitat that were neighboring each field that we could use to try to look at the effect of landscape context. We then, again, use these 3-meter resolution images from the Planet SuperDove imagery and classified the different types of land cover using a random forest algorithm into these different categories. So we had other alfalfa, weeds, which is what we were particularly sort of as kind of semi-natural habitat we thought might be important to refuges for predators, and then agriculture that wasn't alfalfa, bare ground, impermeable surfaces, so-called natural vegetation, which was mostly greasewood dominated landscapes, riparian along those rivers, and also that surface water component, which we thought might be a good predictor of the kinds of ditch effects that we were seeing for flood irrigated alfalfa. So we have just a little here description of how this worked for those who might be less familiar with how you might use this kind of machine learning framework to classify the types of land cover. So we acquired the images. We then classify those images using a random forest, and Anson made me a nice little diagram of how random forest works, which is coming up shortly. Essentially, what he did was he first went into these landscapes and assigned manually what he thought a particular type of land cover was. So for example, this I believe would all be riparian land cover, and this would be an alfalfa field. And that's what he calls the training data. He could then reserve a subset of that training data and use his ultimate random forest predictions to see how well the random forest predictions was predicting what he'd assigned manually. And he found that on average, it was working about 97% of the time. So fairly accurate predictions of the kinds of things that you could see manually using this random forest approach. The kinds of information that we have in these Planet Dove images is reflectance values and various derivatives like vegetation indices like NDVI. And these reflectance values of these different colors, every pixel has these different values, and the random forest can use the value of the pixels in all of these different reflectance values, and their derived indices to try to classify that particular point in space as a particular type of landscape cover. Okay, so this is an example of a decision tree that's used in a random forest. So these particular variables that are used are chosen by the random forest algorithm randomly, and can be chosen multiple times. But what ends up happening is that you get these dichotomous decision trees for each pixel, and it can be something like this for reflectance value, red value. Is it over some particular value? And if it is, then it asks the next question, is it over an even higher value? Or if it's not, it asks another randomly chosen variable, the blue reflectance value, is it over some particular value? And eventually, using a tree that would end up being much more complex than this one, but always these dichotomous branching points, you end up with a classification, predicted classification of these different land cover types that we were interested in. That process then happens 10,000 times, and then you basically use a vote counting majority rules framework for choosing what a particular pixel represents in terms of our what ended up being 8 land cover categories. Yeah, the one important point about this is that this is his last point, is that this process does not require that we choose which spectral information we include. It just chooses from all of those potential reflectance values and their indices randomly to produce these decision trees. Okay, so one of the really interesting things that we learned from this was how different manual digitization could end up being from this random forest method of classifying the landscapes. And one way in particular that this ended up mattering to our study was in terms of weedy plant classification, because of the way because of weeds being omnipresent in these landscapes and alfalfa being a perennial crop and so on and so forth, there is weedy intrusion into alfalfa fields. And so if you were to go and digitize a map of where the alfalfa fields were, you would say that this full gray box is an alfalfa field. Whereas if you use the random forest classifier, you get what this dark gray is, which is a lot more estimate of weeds, weed reflectance values within that field, and that ended up being really important to our results. All right, another really important piece of this was that we were looking at landscapes around the points in the fields that we chose to do our experiments at a 1 kilometer scale. So these are circles of 1 kilometer radius. But we weren't sure what spatial scale was going to be the most relevant to providing refuge habitats for alfalfa aphid predators. And the way that we were able to address this using this kind of landscape classification was to ask, given a set of continuous weighting functions at different distances, what kind of weighting seemed to best predict predator densities and therefore the effects of biocontrol that we were able to see in our experiments. So for example, this red weighting scheme here heavily weights those kinds of land cover that are very close to the field, whereas this purple weighting function would provide more weighting to those types of land cover that are a little bit further from the field. And so you'd be able to see, based on model selection, whether it's those landscape variables very close to the field or some intermediate distance, or those farther from the field within our 1 kilometer circles that were of most importance to determining predator densities. So here's an example of how that would look. So here, this red weighting scheme, which gives the greatest weight to those habitats that are very close to our experimental sites. Basically, the land cover types within this very small circle are the ones that would be predictive or not of predator densities, whereas in this purple weighting scheme, everything in kind of the light white here within our 1 kilometer circle would potentially be predictive of the predators. Just another way of showing that and how this could affect your results. So for example, if we say that this approximately 50% of land cover in our 1 kilometer radius circle around our experimental point is alfalfa. But then we go into one of those weighting schemes that weights the land cover types a little closer to the field more heavily. That actually doesn't, in this case, create that much of a change. About 50% of the land cover type would be alfalfa. But if you move into that closest circle, that red weighting scheme from what I showed you before, that really heavily weights those types of land cover really near the experimental sites, then we're only talking about 25% alfalfa. And so then when you're deciding which variables best predict predator densities, it makes a big difference whether you're saying, well, there's 25% alfalfa in this field, does alfalfa predict predators, or 50% alfalfa, right? So how you the weighting scheme that you use will affect the relative proportions of land cover within the circle, and that can either better explain or explain not as well what the kinds of differences you're finding in predator densities. Hopefully that makes sense. Okay, right, and then we choose these just based on model selection. But okay, so that's a lot of methods. Let's get into some of our results. So our questions started here with, well, how do aphid and predator densities vary seasonally? With aphids, essentially, the densities were somewhat higher in the fall, and that really came down to higher densities of non-pea aphids and blue alfalfa aphids. So basically, those two other genera of aphids, which were the cow pea aphid and the spotted alfalfa aphid, had higher densities in the fall, and that led to this sort of higher tail of aphid densities in the fall. So although the means weren't too different, you generally had slightly higher aphid densities in the fall. And just to highlight how much those densities of cowpea and spotted alfalfa aphids changed between seasons, we had basically no cowpea or spotted alfalfa aphids in the spring, but quite a few in the fall. Whereas again, the pea aphids and blue alfalfa aphids maintained pretty constant densities between the two seasons. So this was in contrast to the predators, which generally had higher densities in the spring. So I should have explained in the last slide that here we're looking at green being the spring distribution and red being the fall distribution. So we have some of these predator taxa that show minor decreases in the fall. So again, that leftward shift of the red curve means that they're found at lower densities in the fall. Some of what ended up being really important predators, two aphids show major decreases in the fall. So you can see the difference in this distribution of ladybugs, for example. There's very close to 0 ladybugs in many plots in the fall, whereas there were several in each plot in the spring. Same for Geochorus and Ichneumonids. And Mavis, really very few in either season. All right, so how do aphid and predator densities vary seasonally? Aphid densities are somewhat higher in the fall, but potentially control is important in the spring as well. We still have fairly high densities of pea aphids and blue alfalfa aphids. There's generally probably better potential for biocontrol in the spring because we have considerable densities of those predators in the spring and not so much in the fall. Okay, so then key to our understanding of the system then is which predators actually suppress aphids, and that's what our experiment was designed to answer. So this ends up being, and the reason that we took this experimental approach, as I briefly explained before, is that it ends up being not such an easy question to answer because of this covariation between predators and prey, right? So this is like a typical type of Lotka-Volterra type model of population dynamics of predators and prey, where you get a general peak of predator densities following a peak in prey densities, and then the prey densities will start to go down as the predators really control them. And as the prey densities go down, the predator densities will also then go down, and et cetera, et cetera. You get these oscillations, right, of predator and prey populations. So if you're just looking at correlations in the densities between predator taxa and prey taxa, or predator taxa and pest taxa, then you could get a little bit lost in these positive correlations and understanding really what's really important to suppressing aphid populations. But first, you need to know which of those do track aphid density, because that's your hypothesized agents of biocontrol. And what we found was that was also variable by season. In the spring, it was anthochorid and coccinella densities that tracked aphid density. And in the fall, it was the wasps, the Ichneumonids, and Geochorus that were tracking aphid density. And this is a map of the variable importance from our models. So basically a measure of how well a particular predator's density can explain the observed variations in aphid density. And so these big red bars just show that that means that ladybugs and anthocorids are very well correlated with aphid densities in the spring and so forth. All right, and this is an example of that positive correlation then between the density of the predator taxa and the density of aphids. And then this is from the fall. Interestingly, ichneumonid still show that positive correlation, which is what you'd predict. And then although the variable importance here for geochorus was high, in fact, there's a negative association between geochorus and aphids. So I don't know what that's about, but it doesn't seem like these are either a very good candidate for aphid suppression. All right, so what we ended up doing then, right, to determine which predators actually had an impact on suppressing aphid populations was to use these, what had been sham treatments, but then became our predator attractor treatments to understand what a given increase in the density of one predator might mean in reducing aphid populations, or whether it was associated with reduced aphid populations when you increase the density of that predator. And so our canopy treatments were effective at attracting some predators in the spring specifically. And these were the 3 predator taxa that we successfully attracted to those canopies. We had higher abundances of Anthocoridae, arachnid spiders, and ladybugs in the spring on our canopy plots than we did in our open control plots. So then we could use those plots where we'd attracted predators to ask whether given amount of increase in predator abundance was associated with a reduction in aphid abundance in that plot. And this was true only, it turned out, for ladybugs. So aphid density is lower in canopy treatments only when coccinella beetles or ladybug beetles are present. So this suggests that among all of those 6 predator taxa that we thought might be important, we can only detect, using our data, an effect of coccinella beetles as an effective biocontrol agent. And this is only in the spring at our sites. These canopy associated increases or experimental associated increases in anthocorids and arachnids were not associated with a decrease in aphids. All right, so then we were really focusing in on coccinella lady beetles as potential agents of biocontrol of these aphids, and interested in then what landscape features might predict higher pest densities and what landscape features might predict higher densities of ladybug beetles, and whether that was occurring in landscape types or land cover types close to the field or far away from the field. So what was the relevant spatial scale to consider? All right, we have more of these variable heat maps coming up, and what these heat maps will show is a measure of the variable importance in the model that we built that incorporated an element of covariation between predators and prey, an element of what site we were working at, as well as all of the different kinds of land covers that we were looking at, and whether we were looking at that weighting of heavily weighting land cover types close to the field or allowing land cover types farther from the field to have an influence. And so in these heat maps, red will indicate variables of high importance, blue will indicate variables of low importance. And along the rows here, we have the different kinds of distance weighting. So I'll just remove this for just a second so you can see it. So this very aggressive distance weighting is what weights the land cover types very close to the field most highly, whereas slight distance weighting weights those land cover types farthest from the field, as well as what's close to the field. And then we also had a constant distance weighting, constant reduction in distance weighting. So the farther you got from the field, you linearly reduced how much you incorporated that land cover type, and then no distance weighting at all, which is what would be just if you just took every land cover type in that full 1 kilometer radius circle and looked at it equally. So, okay, so that's rows. And then columns are our different land cover types. And again, remember, we were particularly interested in these kinds of natural and semi-natural habitats, and potentially in the role of how water influenced plant communities in these communities, whether that might affect the kinds of refuges that were available to these arthropods. And finally, in each of these heat maps, he has an average of all of the other columns, so that helps you, for example, to determine the best spatial scale to be looking at these different land cover types at. All right, so again, getting back to ladybugs, because those are the predators for which we have the best evidence that they're actually suppressing aphid densities. What we found is that ladybugs are positively associated to weedy cover very close to the field. So again, and also negatively associated to bare soil. But in this weedy land cover category, you can see that under this very aggressive distance weighting, so basically weighting those weeds that are very close to our experimental plots most heavily, we get the best prediction of our coccinella densities. And this is just the way you would look at that in regression form. As you get more weedy cover, you get higher ladybug densities. All right, so here we're just showing that this difference in these far ends of the spectrum here, from low weedy cover to high weedy cover, and two of our sites here. So this is Minden Field 1, where there was almost no weedy cover near the sampling location. Again, the sampling location is in the middle of this dark circle, which you can see here, and you'd see something that looked like this blue color if there were weeds, but there isn't anything, and we got very low ladybugs in that field. Whereas this is one of our field experimental plots that had the highest surrounding nearby weedy cover, and that's what that looks like here, as identified by the random forest algorithm from the planet imagery. All right, so one of the things, again, as I alluded to earlier, that this really showed us was how much weeds in the very close proximity to our experimental plots seem to matter. And we would have missed this result had we tried to accomplish this analysis using manual digitization of these maps. Because that random forest algorithm successfully identified these weedy patches that are kind of encroaching into the alfalfa field. And again, because that most aggressive distance weighting is really focused on that area immediately surrounding our experimental plots, those weeds that actually made their way into the field or were right along the ditches that surrounded those fields had the biggest impact then on our models. And so we didn't get, if we looked at nearby weedy cover when we classified alfalfa fields manually, we got a slight reduction in our model fit, but when we produced these larger models that had all our covariates and included site and so forth, weeds didn't actually come out as a factor of importance in predicting coccinella densities. It was only when we used this classification using the reflectance values in these decision trees of the random forest. All right, so in addition to ladybug predators, which we think are important biocontrol agents of aphids being associated with land cover types and particularly with weeds, we also found that aphids were associated with particular land cover features. And again, this was variable between spring and fall. So in the spring, it was plant species richness in our field margins, which these were data that we took on the ground of surveying ditches just around the fields. Flood irrigation ended up having a very positive association with aphid density. So sprinkler irrigated fields had fewer aphids than flood irrigated fields. And again, this positive predator-prey covariation, so we get a positive association between the density of ladybug, coccinella beetles, and aphids. In the fall, again, we have an effect here of field margin plants, and then we had a negative effect of impermeable surfaces and a positive effect of that kind of desert shrub landscape. So in this case, in the fall, it seemed like the more desert shrub you had around of that kind of vegetation type, the more aphids you actually had in the fields. This is just an interesting thing to highlight here, that this species diversity quantified two ways, either as species richness or as Shannon diversity in the field margins, essentially along irrigation ditches, had differing effects on pest densities depending on what season we were looking at. So in the spring, it seemed to have a positive effect, having higher diversity in those ditches, and in the fall, it seemed to have a negative effect. And this is probably due to differences in plant composition in these field margins, which is something that was beyond the scope of what we did here. Another thing that was very clear, that basically led us to that understanding that irrigation method was associated with pest densities was a positive association with surface water that came out of our models. And again, remember that we mapped surface water very carefully using that USGS hydrology data set. And we found, before we had hypothesized that irrigation method was going to be very important, we found this very strong positive association with surface water at the landscape scale here. So remember that these higher rows indicate these distance weightings that take into account that entire circle of land cover types. So at these large spatial scales, total surface water seemed to be a positive predictor of aphid densities. And that was not something to do with the particular vegetation types that were associated with surface water. So it didn't seem to be something driven by vegetation types associated with surface water, but rather something else about surface water. And so we reasoned, as Anson was actually first presenting these data for a conference, we reasoned that surface water should tend to predict where fields are being flood irrigated in these systems. So where we have a lot of canal systems, we tend to have flood irrigation and a lot of surface water. And then in areas that are sprinkler irrigated, we do not have that surface water. And so here you can see that the positive association between surface water and aphid density corresponds to a very strong relationship in watering method and aphid density, which ended up being the better predictor of aphids than just surface water in our models. So we just stuck with this as basically this was a hint that this is what was important to aphid densities, was the irrigation method. Why this is, we don't know. There are hypotheses out there in the ecology literature about how periodic plant water stress can increase nitrogen availability to aphids, and that could be a positive thing for aphid populations, but it could be other things as well that we don't know. Possibly, sprinklers are just too much physical pressure on the aphids on the plants. I think this remains to be looked into, but it was a very strong effect in our data set. All right, so if we put all of this together, then what have we learned about the kinds of land cover types that affect aphids as well as their potential biocontrol agents? And specifically now we're talking about ladybug beetles, because that's where we could detect an effect of pest suppression by a predator. What we did to evaluate the entire model of what might be going on is some structural equation modeling, where we put in our variables that ended up being of highest importance from our overall models, and asked how much they comparatively could explain pest densities in our system. So again, this is kind of results from the aphid density graph that I showed you earlier. There's a positive effect of flood irrigation on aphid densities, and in the spring, a positive effect of plant species richness in the field margins on aphid densities. And again, we don't know exactly what this means. We suspect it means that there are alternative aphid hosts in those field margins in the spring. Then look at the negative effect here of canopy treatment. So where we added through attraction of ladybug beetles to those nettings on top of our plots, we get a very strong suppressive effect relative to either of these positive effects that we found of land cover types. We get a very strong suppressive effect of ladybug beetles on aphid densities. And now we can then connect to what kinds of land cover types affect ladybugs, which we know in the spring is a positive effect of that nearby weedy cover and a negative effect of bare soil, such that we calculate, putting all of this together, that weedy cover effect of increasing ladybug beetle densities has a comparably negative effect on total aphid densities to that of the positive effect of flood irrigation. So those are kind of rivaling effects in their strength on your resulting aphid density in those fields. In the fall, we couldn't detect any effective biocontrol on aphids. Again, it was the ichneumon parasitoid wasps that had the most strong positive association with aphids in the plots, but we couldn't detect a reduction in aphid densities on those plots where we had an increase in parasitoid wasps. So we're kind of without a hypothesized biocontrol agent, but we do have these different direct effects on aphids in the fall, where impermeable surfaces seem to have a negative suppressive effect on aphids, and desert shrub, somewhat surprisingly, at least to me, seems to have a positive effect on aphid densities in the fall. And remember that those aphid densities are strongly driven in the fall by cowpea aphids and spotted alfalfa aphids. All right, so our takeaways then from this overall analysis are that these beneficial arthropods likely need in these periodically disturbed systems some sort of refuge habitat to establish and persist, and that our methodology here of using remote sensing and using those remotely sensed images and random forest to identify what kind of land cover was in each pixel in those images can help us better identify potential refuges than we were doing on our own by manually digitizing those photographs. We can also use these kinds of constant or continuous, rather, distance weightings to understand at what spatial scale might be the most important for maintaining these kinds of refuges. And again, in the spring especially, it was these very close spatial scales, these very nearby to the field ones that were the most important. And those, spring is when we were able to identify biocontrol. So what we can say for biocontrol in the system is it seems to be possible in the spring and mediated by these land cover types that are very close to the field. So overall then, ladybugs and weedy cover seem to be good for pest management of aphids in these systems. But it must be noted here, right, that having weeds next to your field and particularly in your field can also be a bad thing for hay quality. So it really remains to be evaluated. So how much you could sort of mitigate the negative effects of weeds in the hay, and use weeds as a positive driver of predator densities without having too much weed incursion in the field. Flood irrigation seems to be bad for pest densities, or basically it encourages those pests in ways that we did not initially hypothesize. And so that, again, the mechanisms of that I think are worth looking into. But for the moment, we can say that in our system, flood irrigation seems to be associated with higher pest densities and no impact of that of biocontrol. So you just have higher pests. And with that, I will thank the funding agency. This has been a great project to work on. Everybody who worked on it, and Steve Foster, who was working on with me in University of Nevada Extension to talk to our area farmers about this research and its results, and all of you for attending. And I'd be happy to try to answer any of your questions.
All right, Matt Bauer has posted the first question in the meeting chat, so we'll get started with his. And it says, I'm wondering how you might model the costs and benefits associated with weed presence in the spring. Weedy borders mean higher predators, but they also mean weed seeds and potential reservoirs of plant pathogens that may impact alfalfa.
Yeah, absolutely. And we need another experiment. We can't do it with our present data. But I think you'd have to get further into yield and yield quality, I think, to be able to model that effectively, which we haven't done here. But I think it's certainly worth looking into, and particularly the extent to which possibly using our data, we could try to exclude the data to just those weeds that are at the border of the field rather than the weedy incursion, and see if you could use just those weeds to try to actually get an increase in the densities of biocontrol predators, because you would think that those weeds would be less bad for the overall hay quality than the ones that actually get into the fields. So we could possibly do that, but then I think beyond that, we really have to see more about the plant composition of these borders and more about what kinds of weeds are better at getting into the crops themselves and doing harm to the hay quality.
And so we got a couple more questions. This is good. What's the next step to exploring creating refuges for predators in these fields? Will you explore hedgerows or vegetation strips designed specifically for the desert environment?
Yes, I think we could do that. My attempts so far to generate some manipulative experiments on weedy hedgerows or vegetation strips have failed. I have done a terrible job at basically overcoming the natural weed pressure in the experimental fields that I have at my disposal to try to manipulate the weeds to what I want them to be. But I think that it's still a fruitful area for research, maybe with someone with a better green thumb than I, although I've had several undergraduates over the past few years trying to help me with this, and we've all failed. But yeah, I think we should be doing, some manipulative experiments to try to get in there and see these different plant compositions. What we initially hypothesized was things that were not legumes, essentially things that were not closely related to alfalfa, but that did have aphid resources or floral resources that we think would be good for predators, would be particularly good weeds as far as biocontrol go for the aphids. And then the next question would be how much do those disperse and so on. So how much are they correspondingly negative or neutral for the weed management part of it.
Yeah. And then another one, Thomas gets generally, what were the species of weeds on the field margin? Did any of the fields have desirable species on the field margin, like introduced perennial wheatgrass? Um, what would be, what do you think would be the impact to lady beetle populations in perennial grass vegetation compared to weeds, compared to bare ground?
Yeah, that's an excellent question to which I do not have the answer. But I'm happy to follow up with our data. We did try to identify all of these weeds. So we should know more about that than I do currently. My general impression is that in our system, the aphid populations are more dense on forbs than on grass. And so I don't think that grasses necessarily, if we're looking at alternative aphid sources, would be good for aphid predator populations. But that's just my anecdotal gut feeling, and I'd be happy to follow up with you about anything that's actually coming out of our data about that question.
All right. I had one about the satellites. You said that those images were $50. So at each study location, how many $50 images did you need each time? And how frequently did you have new imagery taken?
So the plant dove is trying to photograph the entire surface of the world every day. And they haven't gotten there yet. So currently they're doing it every 2 weeks. And we chose a single image for each field that was based on essentially what the cloud cover characteristics were on that day. So we tried to get the clearest image. We tried to make sure that the alfalfa fields hadn't been recently harvested, so that was constant across all of our images, using reflectance values which would have indicated whether alfalfa fields were high or low. And then the days with the least cloud cover, and then we just purchased a single image that depicted all of those things for a given day. So I think the total cost of our imagery was $150 for this project, or $200, one for each site.
That's amazing. Okay. Do you have any work on the effect of various insecticides used for aphids, weevils on beneficial populations?
We do not. And I don't know if my colleague, Ayman Mustafa, who's in Arizona, who's been working more on insecticides and aphids, he might have more of those data, but we haven't been working on insecticides.
Okay. Any other questions? Anybody want to type into the chat or come off mute and ask? If not, Beth, thank you very much. That was a great presentation. And I want to tease the next one, which will be Rebecca Schmidt from USDA ARS. And in the biocontrol realm, she's going to be talking about augmented biocontrol on Pacific Northwest tree fruit. That's October 11th at noon. We do have more comments, which is good. Thomas says, up in Susanville, we have lots of broadleaf weed species and annual weed species surrounding fields where perennial grasses are not established on the margins. These are typically undesirable plants and are weedy in the stands themselves. And thanks for the presentation. Yeah.
My pleasure.
All right. Well, thank you, everyone. Again, thank you, Beth. Really appreciate it. It was a great presentation, and thanks for tuning in. And we'll see you all next time.